So in this video, we're going to discuss the two things, the sources of that NADPH that we were talking about in fatty acid synthesis, as well as the source of the acetyl-CoAs for fatty acid synthesis. As we'll find out, for one of the processes, or one of the sources of NADPH is actually attached to those sources of the acetyl-CoAs. So kind of makes sense. All these things are connected in some way or another. Okay, so two main sources for NADPH. The bigger one, or the one that produces more NADPH per molecule, is going to be the pentose phosphate pathway. Okay, and that's going from glucose 6-phosphate to ribulose 5-phosphate. And in that process, we're going to make two NADPHs. Okay, but we'll talk about that more in another video. Okay, the other one that we're going to talk about is going to be used in concert with the production of these acetyl-CoAs. Alright, specifically, and we'll talk about it now, we'll go more in depth later. We're going to have malate being turned into pyruvate, and in doing pyruvate and by kicking off a CO2. And in doing so, we're actually going to reduce an NADP plus to an NADPH with an associated hydronium ion, the H plus. Alright, so where the hell did that come from? Okay, well, this is actually going to be borrowing in part from the TCA cycle. Okay, so we need our sources of acetyl-CoAs is going to come from the TCA cycle. But remember, where is that happening? That's right, it's happening in the mitochondria. But fatty acid synthesis is happening in the cytosol. So we need to get those acetyl-CoAs that are currently in the mitochondria and move them back out to the cytosol. How are we going to do that? Well, if you look at the picture, we're going to start out with the top left with citrate. Alright, it's going to go through a citrate transporter and then go through move through the inner membrane, out through the outer membrane, and from here we're actually going to break apart the citrates. We're going to break off 2 carbons, and we're going to create an oxaloacetate and an acetyl-CoA. To do this, we need energy. So I was actually lying to you in my first video, okay? It's not 1 ATP per elongation, because technically this is the second ATP, alright? So the actual source of acetyl-CoAs needs ATP, so it's actually 2 ATP per, but probably not that big of a deal. Anyway, after citrate has been transformed into oxaloacetate, we're actually going to use malate dehydrogenase to transform that oxaloacetate to a malate. Alright? To do so, we're going to reduce the oxaloacetate using NADH to produce malate. Now here, malate can do one of two things. It can either go back into the mitochondria via the malate alpha-ketoglutarate transporter, or it can be complexed with what's called the malic enzyme. Alright? The malic enzyme is going to kick off a carbon group in the form of CO2, and in doing so, it's actually going to reduce that NADP plus into NADPH. Alright? So then we have malate, it changes back into pyruvate, and then kind of think of just what pyruvate does. Alright, it's going to just shuttle back into the mitochondria, and from here it can do one of two things. It can either go back into that pyruvate dehydrogenase complex, get turned into acetyl-CoA, or we can actually add a CO2 and using the energy of ATP, transform that pyruvate into oxaloacetate. Alright, so not only do we talk about where we get our acetyl-CoAs, changing that citrate into an oxaloacetate by using ATP, but we also talked about where we got that NADPH, alright, and that was changing the malate into a pyruvate molecule. Okay, cool.